So maybe I can at least start to introduce our speaker for today. So as you know, we are in this last part of the course where we discuss about evolutionary topics. And so this is, we still have 3 lessons to go. Today, we have Gianni Liti from IRCAN in France, in Nice. So Gianni got his PhD at the University of Perugia in biology. Then he moved to England for his postdoc in Nottingham, in Leicester, if I understand. And he has always been working with yeast on evolutionary topics. And then he moved from there to CNRS in France, where he is now, where he leads the group on population genomics and complex traits. So Gianni has always been interested in issues related with evolutionary and quantitative genomics. And this, I guess, is what he will tell us about today. So please, Gianni, I leave it to you.
Yeah, so thanks, Andrea, for the kind invitation. It's a pleasure to give this lecture today. So our team in Nice work on population genomics and complex traits, mostly using barley yeast as a model system. And so I organize this lecture with an introductory part, like Andrea suggested. And then on the second part, I want to show a couple of examples of projects that I've been working for several years that sort of derive from the first part, from the first introductory part, which is about genome evolution across timescales. So we're interested in understanding, but also measuring genome evolution. This can occur across timescales, and this can have implication for many fields, like for medicine or also for agriculture, to improve crops, as well as to understand how species evolve. So I added some examples in the first part, mostly related to human, where you might be more familiar. And I started with illustrating where we are with the human genome. So the human reference genome has been a goal for many years, and has been a common effort across continents and countries. And it was first released in the early 2000, and this has been continuously updated. So a few weeks ago, the Telomere to Telomere Consortium has released the most up-to-date version of the human reference genomes, which consists of approximately of 3 gigabases. And this is the comparison with the previous version, the GRCH38. It was released 3 years earlier. And essentially, we're now moving very close to essentially a gapless genome. So where all the nucleotide information that constitute the blueprint, the genetic information, is contiguous and organized in the 23 linear chromosomes. And so there is basically there are no more gaps or unplaced spaces. And essentially, this is mostly related to difficult area of the genomes, like around centromeres, where there are many repeats. And as this figure is showing, and this is where most of the gaps have been fixed. And so nowadays we have nearly completed applied version of the human reference, from which we can derive information, like, for example, the ancestry, so where the origin of these segments that were missing in the original release or the previous release. So the genetic information of a genome is organized in genetic units, and again, this is just comparing the latest release of a couple of weeks ago in terms of genetic units. So there are about 20,000 genes. So this number has not changed much, because this was sort of the easiest part to decode in a sense. So this has been less interested by this new update. And there are around 60,000 genes, of which 20 they do code for proteins, others they do code mostly for other type of transcripts. And this is what it will translate. So these are the genetic information we translate into sort of genetic information that make a species, in this case, the human species. But what about genetic variation? So we know that we accumulate variation over time, and essentially these are largely named as mutations. So this mutation can be defined as alteration of a permanent alteration of a nucleotide sequence. And normally these occur through cell cycle or through some sort of maintenance of the genomes, like for example during DNA repair. So this can occur spontaneously during, for example, the cell cycle. But also there are conditions, like for example chemical condition that can induce and increase the rate of mutations. So how do we detect that? Well, we just said before, we have the reference genome. We sort of provide an infrastructure to map how the genome evolves. So let's say we have a reference genome, and somehow this has acquired mutations. So this will accumulate, for example, a single nucleotide mutation at this level. So this is one way that genomes evolve, and these are referred as point mutation, which their actual frequency can vary through space, so they can accumulate different rates depending on what are their position in the genomes. But also there are larger type of events. So these are generally referred as a structural variation, and the sort of cutoff that is set is around 50 base pairs, although this is arbitrary. So also these come with different flavors. So again, for detecting them, we usually use a reference genome. And in this case, for example, reference genome, it does have a segment that is now present in our sample. So this is a deletion. The reverse is an insertion in our sample compared to the reference genome. For example, it could be related to a mobile element inserted in this place. Also, there are other type of things that can change copy number. For example, we can have duplication of a gene, so the reference is a single copy, whereas this is 2 units that can be next to each other or can be interspersed in the genome. Or there are other things, for example, like inversion or translocation, which in this case, they do not necessarily change the copy number, but they change the order of the genetic units that are contained in these segments. So the accumulation of mutation, it does occur through time, and in most cases, so we measure these times in terms of generations. So we can have, in a way, we can have 2 types of generations. So we can have generation referred as sort of mitotic or somatic, which they could produce mutation, with the mutation accumulate. Also for many organisms, including us, that are obligate sexual. So these also, there is a mutation that can accumulate through the gamete formation, and these are passed through the offsprings. And as we said before, the point mutation or the other type of structural variation, the large scale, that can occur at drastically different rates, so several order magnitude of difference. And usually point mutation occur at very low frequency, but they occur, and given the population size in this case will be the cell numbers, they occur and they propagate through different generations. So for the somatic mutations, so we talk about somatic mutation in human, for example, the one that accumulate through the development of an individual, and possibly I think the best known type of such mutation are the one that occur during tumor formation. So there are many mutations that that being characterized actually to be driving the development of the disease. And usually what happens in this case to track them and to understand how genome evolution occurred through cancer development, there are approach where basically there are sequencing of the cancer samples through space and times, and these usually are matched to sort of a normal sample genome. It's known that this such genetic mosaicism it can also affect aging, and given the fact that there is such genetic heterogeneity, these are difficult. Such heterogeneity is difficult to describe and to capture and to quantify. So there have been both experimental and computational approaches to sort of deconvolute such heterogeneity. So we are also sort of mosaic in a sense that our genome derived from the one of our parents, and this sort of, so the 23 chromosome we did see in the reference genome, actually it's sort of a simplification of a real human genome, so which is composed of 23 pairs. And in this case, actually what can be done also, the diploid genomes can be phased, so the haplotype can be resolved, and the chromosome can be binned into two different pair of the chromosomes. So this can be done in different ways. For example, previous approaches have used like family type of triogenomics, where both the parents and the offspring sequence, and this will enable to sort of bin genome into the correct yellow and blue haplotypes. But also, again, new methods using long reads and computational approaches that are enabled to do such phasing without the usage of parental information. This also is a result of meiotic recombination. So during the production of gametes, if we do produce gametes from this one, the haploid genome of the gametes, it would be recombined yellow and blue blocks genomes. In addition, also there will be between 30 to 80 new mutations in the gametes, depending on if it's the sperm, and this will again constitute new mutation, new variants compared to what has been inherited from the parents. So again, we can now move to a longer time scale. For example, if we look at variation, which again, these were formed by mutation over times that are segregating in the human populations. We can capture that, for example, by population genomics. So this is one of the latest studies showing about human genetic variation across 1,000 genomes that were sampled through the different continents. And basically, a couple of things I would like to point out here is it's the total variation. So the total variation in terms of genetic variants that are present in this population. So one thing that captures sort of the human evolution is this is more abundant in Africa, reflecting the origin of the human species and the subsequent out of Africa that have generated, that have dispersed the human population through the globe. So this results on higher amount of genetic variation and private to this population. But another thing that's emerged through population genomics is that human, after have left Africa, they also have mixed with archaic species like Neanderthal or Denisova. And this is depleted in Africa because this has occurred after the out of Africa event, and for example, European population have some ancestry around 2% of the genomes that has Neanderthal origin, and there were other species like the Denisova that have been detected in, especially in the population of Asia and Oceania. So we can further step back, and essentially we can also compare our genomes. So this is a couple of human genomes from different populations that have been compared between them, but then they've been sort of step back and compared with the other great ape genomes that have diverged between 10 to 15 million years ago. And these are the sort of number of events in terms of structural variation. So in this specific study, actually, they produce high quality genomes for several of these great apes. So to compare the accumulation of structural variation, which these are the events in terms of deletion, insertion, and inversions. And this is the amount of DNA that is interested in this event. So this ultimately they do have an effect, and have an effect in terms of phenotypes. And this is true also for the point mutations. So what we do see, it's not just the results of the rate at which these different events occur, but also they are exposed to selection. And this has been intensively studied by evolutionary biology, where there's been different models, different selection model that will have predicted a sort of fitness effect of different type of mutations. And for example, in a sort of a neutral evolutionary model, we will expect that most of the new mutation will come out and will be deleterious. But this might be actually becoming, part of it become beneficial when these are exposed to selection. And in this panel, actually, this is one study from Yetwas. It's a bit old, but it's very nice, where he did try to assess experimentally the sort of the fitness effect of mutation in a specific segment of a gene, of a chaperone in this case. And actually does match sort of the expectation, where many of the mutation, in this case is a mutation will change the amino acid sequence, will result in a sort of deleterious changes, and very few will be the one above the zero, above this zero on the selection coefficient. It will be sort of being advantageous, and in this gray line, it's actually mutations that are not change amino acid sequence. So many of them, they have no effect, and doesn't seem to be any detectable deleterious effect, with few that seems to be beneficial. So this bottom panel is being done in yeast, which is what we study, and on the next set of slides, I'm going to show a little bit of why yeast has become sort of a primary model in biology and specifically in genomics. So yeast actually have been used in biology, but actually their initial usage by human in terms of also understanding the biology it comes from industry. So these were the first sort of the birth of yeast genetics, and this was done in a lab that was associated with the Carlsberg in the Carlsberg Institute for Beer Production. So these were actually gametes were dissected into agar plates and of strains were used in fermentation, and actually also this media it's media that is used to study fermentation. So there's been a strong connection between actually the usage of yeast for industry and its biology. So yeast are fungi, in this case are single cell eukaryotes. So they sort of developed these such single cell states by losing multicellularity multiple times, and there are many hundreds of species. And just to give a nice overview, visual overview of the diversity of yeast. So these are body. So it's by forming daughter cells and bud, and this is sort of their genetic distance inferred through amino acid substitution rates. And basically, you see that also within just body yeast, we do have a huge evolution span compared to animals. For example, comparing human to other animals or plants like Arabidopsis thaliana, which is one of the classic model system, to other plants. So in our lab, actually, we mostly work with this group of yeast, which in terms of let's say evolution space, only sample a small fraction of the yeast diversity, in this case, the body yeast diversity, but still this is comparable to maybe what we look in terms of evolution between human and mouse.
So where would Pombi be in this plot?
So Pombi probably would be, it would be much further away. So
the difference between Pombi and budding yeast is like between human and roundworm.
Yeah, it will be even further. So it would be very hard to place here. Mm-hmm. It's really deep.
Interesting.
So one thing that we actually are interested in is the human yeast interaction, and as I will show you later on my presentation, human had quite an important part in shaping the body yeast evolution, and this is just some of the early evidence of winemaking, and this around 8,000 years ago in Georgia, where the early evidence of winemaking, and there are some even older in China in terms of beer making, and which date around 10,000 years ago. Just to say that basically human has been exploiting yeast for food production, especially for fermented food and beverage, for nearly 10,000 years. And as you will see, this had a tremendous impact on its genome evolution. So the other reason why yeast sort of took a prominent role in studying genetics and genomics is essentially because we have a strict control of its cell cycle. So we can isolate sort of single cells and manipulate them. And for example, we can go through sexual phase. So this would be sort of similar to the gametes formation that we've seen before. Human as well, we can take single cell physically isolate them and propagate them only to form sort of this colony and, for example, study genetic heterogeneity there. So this has been a powerful advantage of budding yeast in developing new approaches in genetics and genomics. In fact, budding yeast is definitely been a prominent model in genomics. So it was the first eukaryote essentially to be used to develop large-scale genomics projects. So and a lot of this is due to the fact that its genome is very small, as we'll see later, but essentially there's been a number of breakthroughs in yeast genomics where, for example, the first chromosome of a eukaryote was sequenced in 1992, so this was chromosome 3. It's not the smallest, but it's a small one, but it's a special because it does actually contain the methane type, basically the sexual information of the budding yeast. So this was a lot of manual work and through sort of different type of libraries and sort of reading sequence and then designing the approach to sequence the next one. And it took a couple of years to do it. And eventually this provided the sequence, and the sequence that was used to sort of also develop approaches to annotate the functional elements. So just to show a few examples, so the chromosome 3 has a centromere, so yeast have a point centromere, so these are smaller, much less complex compared to the human large centromeres. And there was a systematic name given based on their position compared to the centromeres. So this was a progressive name of with the C referring to chromosome 3. So it was the third letter, and these were the gene names. Another feature that he did emerge was the genome of yeast is densely packed. So these sort of arrows are all genes, and you see there is very little space in between them, and this is where the mat locus is with the sex information. So such density sort of fades a little bit out when we get to subtelomeres, which are highly dynamic, as you will see later, and contain a lower gene density and terminate with specific repeats, which help to cap the chromosome ends. So this evolved essentially is chromosome 3 was already part of a worldwide consortium that sequenced all the genome and came out with a landmark paper in 1996. So it was the first eukaryote to be sequenced. And this was a very collaborative effort between mostly European laboratories, but also there were labs in the US and Japan that contribute to this. And release essentially the linear sequence, almost gapless and complete of the laboratory strains called S2SC. So some numbers like what we did see for humans. So we talk about 12 megabase, so it's considerably smaller than what we've seen before. There are about 6,000 genes, which about 5,800 they are protein coding. There are very few introns compared to vertebrate genomes. So most genes they don't have the most completely coding sequence. There are elements like mobile elements. So again, these are present, but much fewer compared to vertebrate genomes. And both centromeres and telomeres have been sort of resolved. Subtelomeric were more complex to resolve than centromere in this case. Two other things that emerged through its analysis. One, again, landmark paper from Ken Wolf study. It did show actually or at that stage we proposed, but then it's been confirmed that the Saccharomyces cerevisiae genome derived from an ancient genome duplication, and this was inferred through the analysis of a duplicated block that were still present in the nowadays genome, and in basically by looking at the gene order within the blocks and also the respective direction compared to the centromere. And so despite most of its genomes, its duplicate genomes has been deleted, so these blocks have sort of supported the whole genome duplication event. There is a reference genome database, so this been also again a leading example. So now to release genomic data information, and also this has been enriched by many of the functional studies and multi-omic studies. So with the development of high throughput sequencing technology, it was possible also to start to sequence closely related species. So in this study from Kellis and Lander, they sequence the sort of the Saccharomyces species complex, which is at that time was comprised of 5 species, and which evolved around 18 million years ago. And by looking at actually, so despite the sequence divergence is quite high when they look at the gene order, it was surprisingly stable. So essentially, this gene 10 in Cerevisiae and 8 and 9 in this case is there were larger steps. So in this case, there is 9 is specific to Cerevisiae, but largely you see that the synthesis, so the gene order conservation it was very stable. Whereas the sequence was highly diverged. So this was in a stretch of nucleotide. You see that the more close you are to Cerevisiae, the less nucleotide divergence you have. So these are ordered through nucleotide, and when you arrive to this species called Saccharomyces bayanus, these start to be highly diverged with many polymorphisms. And looking this at the genome scale, actually, this has enabled sort of studies like phylogenetic footprint. So this is sort of in these tracks there is a sequence conservation across the species. These are the genetic units, are genes, and black you see between genes, this sort of conservation, it does decay. So this has been exploited actually to try to understand the functional elements, and basically does indicate sort of evolution constraints of accumulating genetic mutations. So now I'll tell you a bit more of what we actually do. I'm going to show a couple of examples. So one is how we be using genomics through to long evolutionary timescales to understand its history and evolution. And I'll talk a bit about how to use instead more recent genetic variation to illustrate the genotype to phenotype map. So most of our studies been focused on sequencing the Saccharomyces cerevisiae and the Saccharomyces paradoxus, which is the closest known relative. It's been split from Cerevisiae around 3 to 5 million years ago. And one feature has not been largely associated with human activity. So actually both species in the wild live in trees like this one, which is an oak tree, and sometimes both species can be isolated from the same tree. But in addition to that, Cerevisiae is being then sort of exploited a number of human-based process, and so it can be found in lots of agricultural process that are used in human to produce, for example, fermented beverage and food. So in one of our early study, we sequenced about 40 genomes from Saccharomyces cerevisiae and 40 from Paradoxus, coming from different either geographic place, like for example, North America or from Asia, and or from a specific ecological or domestic niche, like for example, wine or sake. And what we did identify at that time, there were, so we could see what we call clean lineage. So there were lineage specific of these, for example, in this case, a specific winemaking condition, where most of the genetic variation present in this clade was present only in this clade, in the other, what we call clean lineage. But also we did see a number of strains which are this one in these long branches where they had sort of an unmixed ancestry. And to illustrate this, we did sort of plot the same data sets using this software called Unmix tool, and basically you see that most of genetic variation in the one European, in this case, and it's different from the one to the Malaysian isolates, which is this one yellow. But if you take these strains, which would be the strains here, which are mosaic, so part of their ancestry is derived from the wine strains in this case, and part from the sake, and some at the time which we didn't know their origin. And one interesting contrast between cerevisiae and Paradoxus is that if
sorry, Jan, I interrupt you a second. So what are you plotting here on the y-axis? I can't read well. Yeah,
sorry. So these are the fraction of markers assigned to each clade. So this software sort of try to identify a number of population. Usually when you run them, you run the software with a varying number of population and try to identify the population and assign this marker to colors. And in this case, basically assign the blue color to the one European population and these other colors. And essentially what he does, when you get to these sort of mosaic strains, he does see that part of the markers of which are SNPs essentially, they are similar to the one that he identified here in the size.
So they compete. So by definition, if one is 100%, there are no other markers there. This is.
Yes, I thought some colors here, like this sort of orange or purple. Okay. We don't have sort of clean lineage that are, let's say, all purple. So we don't know where this one came from. I mean, rerunning this now with much more genomes, we sort of can trace most of these markers to a specific lineage. At that time, we have a fraction of markers that we could not really identify. But I think what is important here is to see the sort of the structure of the genetic information. This is sort of very homogeneous here, and it's more it's more striking when we compare to Paradoxus. So this is essentially, this is feeding the data without telling the software where the strains are coming from or where they are in the tree. I mean, then we order the strains based on that, but essentially all the genetic variation identified in this block of strains of Paradoxus, they're all very homogeneous, and these strains all come from Europe, essentially. So he was able at that stage to determine that this genetic variation present in these isolates is homogeneous in the European populations. So essentially, you do this also in addition to other type of analysis, like for example, phylogenetic tree or sort of similarity plot. So it's one of the ways to try to understand the ancestry of a genome, or other things like PCA, and so it's one of the tools essentially that we use.
So meaning Paradoxus was very clearly.
Yeah, Paradoxus instead. So essentially, if we take Paradoxus strain and sequence it today, we can clearly say where it is from. And actually, we now sequence hundreds of paradoxus genomes, and there is sort of a gradient, so we can probably say whether this comes from Portugal or Italy or UK and so on. So it's very gradual, it's genetic variation, and there is sort of a geographic gradient that is reflected in the genome. With Cerevisiae, essentially, it's been moved a lot by human, and this is one thing, but also we really think this sort of a mixture as well has been driven by human activities. And so human has done two things. One, move things around, and probably I think we cannot confidently say this is the cause, but I think the fact that the population size in human-related processes is many order of magnitude higher than what are the population size of the yeast in the wild. I think this has contributed to the mixture, and then probably there's been selection for specific breeds.
Okay.
So in addition to looking at the genome, we also look at phenotypes, and again, this was on the previous set of about the Cerevisiae and the paradox were run by sort of where we run growth rates across many hundreds of conditions, and essentially, so one thing that was striking is that, for example, all the paradoxes they group together that tend to have lower phenotypic variation compared to Saccharomyces cerevisiae. So this was a bit paradoxical because in contrast, we didn't see that actually the Paradoxus has more genetic variation in terms of single nucleotide variations, but than Cerevisiae, where with Cerevisiae split in two major groups, but essentially this branch length highness, it does reflect the phenotypic variability. It's true that all the Paradoxus came from the same environment, so they all pretty much all came from these oak trees, whereas the Cerevisiae, they come from many different environments. So this could reflect also the fact that they are phenotypically more variable. And I think now we know why this is the case, and I will show you later. So actually, when Anders Bergström was in my lab, also, we resequenced a number of these strains with the next generation sequencing technology. So we had high coverage, which we didn't have at the time. And what he was able also to do de novo assembly and compare the strains by genome content, which mean whether a single strains compared to another has the same set of genes. So this is the number of gene difference in terms of, or this is the genome content in terms of KB. And this basically was comparing each isolate to each other within the species. So here is very clear to show that, to see that the Paradoxus isolates, which are this yellow one, they vary a lot in terms of SNPs. So, and this is due to actually comparing different subpopulations. So this is comparing European versus American isolates, and this is comparing essentially European versus Far East isolates. So these are the geographic cluster. But when we look at the genome content variation, this didn't scale up with the sequence. Compare instead to Cerevisiae, where we see they are more close in terms of SNPs, but they vary a lot in terms of gene content. So at that stage in 2014, we suggested probably this gene content variation does play a big role in the phenotypic variation. So sequencing technologies evolve, as we've seen also for the human genomes nowadays, but also, again, yeast was one of the first to be used to reconstruct whole genomes. So Yashin Yue was a postdoc in the lab, and essentially he was able to, using long reads, to completely assemble about 12 genomes, five representative of each of the main lineage of Paradoxus and 7 Cerevisiae, and essentially produce complete chromosomes. And in addition to looking at the sequence variation, we're able also to look at the structural variation. And we also use such complete genomes to have a phylogenetic framework where we could base the different evolutionary events. So, for example, these are dot plot metrics of Paradoxus different lineages. So this is the European, which is here, and this is are the South American, North American, European, and sorry, Hawaiian Paradoxus. So we do see an inversions here, and because this inversion is present in all these three strains and not here, we can infer this as at some point here. So the series will have the same orientation of paradoxes, where we can put this here and not here. But then you see that also there is this complex set of inversions in this South American Paradoxus, which is absent in in the North American and Hawaiian Paradoxus, and so basically we can say this has occurred here, and actually this has reflected throughout the genome. So this is the South American Paradoxus. The color reflects the source of ancestral genome structure shared between Cerevisiae and Paradoxus, and we do see that the number of lineage both for Paradoxus and Cerevisiae, they underwent massive genomic instability, and we still don't understand why. So a classic example is this Malaysian Cerevisiae, where, for example, chromosome 7 of the ancestral sequence of all the Cerevisiae is this one, and this has been broken up and now it's split in rearranged in 4 different chromosomes. So we over the time collect larger collection of strains, and we're able to sequence them. So we sequence about we start this 1002 East Genome Project, and ended up with about 1011 genomes, which are arrayed in a single plate, which is what we use to run the phenotypes. So this is the phylogenetic tree of the 1011 strains, which were sort of grouped in around 26 clades. So these are the wine strains, which is the largest number of the clade with largest number of strains, about a third of the collection mapped to this clade, and these are clades that also are related to human activity, as is indicated by this pie chart on the top. Here it does indicate whether the red one are used by human in domestic environments, or these green ones are instead of wild isolates. So you see that here there are many strains used in domestic environments, whereas these ones were mostly the wild lineage. And what it did appear was that we had some highly diverged lineage, and this turned out to be lineage that were isolated in China and more generally in Southeast Asia, which have very high sequence divergence. So through population genomics, actually we're able to show that that Southeast Asia, Taiwan, China are the birthplace of the body yeast. So this has been also recently reconfirmed by both Feng Lianbai lab and Jason Tsai lab, showing this highly diverse Southeast Asia lineage. And in our case, what we did show is that the rest of the non-Asian lineage, they are equally distant to the split with the Paradoxus and with the Chinese isolates, and we propose a single out of China event that funded the rest of the worldwide population. Also, like we did see in before for human, for the African population, we do see a depletion of introgression from Paradoxus. These are secondary contact and are mixed with other species, in this case with Paradoxus. that are depleted in the highly diverged lineage, suggesting that such a mixture occur after the out of China event. So Matteo in my lab has been working on trying to assemble the species pangenome, which is the collection of the genes present at the species level. And basically, this was done again through the novel assembly of these 1011 genomes and to sort of a phylogenetic approach. So for each gene that we de novo assemble from each strains, and we have a collection of around 8,000 genes. So we had 2,000 genes for which they were not present in the reference, for which we did try to understand what they were for. So basically, we found about 1,300 genes that were what we call ancestral segregating. So these were present in all the Saccharomyces species, but their presence absence segregating different strains. So some strains have it and some don't. But if you draw the phylogeny of them with the outgroups, and you will recapitulate the same phylogeny. So these have been evolving together with the rest of the genome for a long time. And these are enriched in some telomeres. Then we do see about 1,000 genes coming from mostly from Saccharomyces paradoxus. So these are interspecies introgression of materials from Saccharomyces paradoxus that has introgressed into Saccharomyces cerevisiae. And finally, we do see a number of genes, about 200, that actually came from highly diverged species. So this we call horizontal gene transfer, and I will show you why we think this one are different from the integration itself, and possibly also the mechanism how they actually enter the Saccharomyces cerevisiae genome is different, but we don't have a definitive mechanism for that how they've been formed.
A definition of introgression?
Yeah, so the definition of introgression, I can go on. So these are introgressed gene from Paradoxus. So this is one of the cerevisiae chromosome. It's chromosome 2. So this is the centromere, and the blue is essentially is a Saccharomyces cerevisiae material. So these are the introgressed block from Saccharomyces paradoxus. Now, if you look at the genes present in these red blocks, they are the same, they are the ortholog of the one of cerevisiae. So this is sort of indicates that there was an hybrid between cerevisiae and paradoxus, and there's been a homologous recombination between the two genomes that have essentially replaced the cerevisiae counterpart. So let's say if you have a gene A within this block, you still have the gene A, but yes, now the paradoxus gene A.
Yeah.
So whereas for the HGT, there is no replacement of genetic material. There is an additional material that largely has come and has been added to the telomere of the Saccharomyces cerevisiae chromosomes.
By gene transfer?
By horizontal gene transfer. So now we don't know if, so one of the classic example is Torula spora, horizontal gene transfer into one yeast of cerevisiae. So we know that if you try in the lab to make hybrids between cerevisiae and Torula spora, these are not viable. So now there can be two things. It can be like uptake of naked DNA, similar to, I don't know, bacterial horizontal gene transfer, and sort of that can be naturally competent to uptake DNA. Or there could be transient hybridization that we've not been able to capture so far in the lab, that will create sort of a transient hybrid, maybe just, I don't know, at the level of the cytoplasm, but enough to, I don't know, to pass some genetic material and to get integrated.
Mm-hmm.
So we don't know that. We're trying to do the experiments to see which, I actually did my PhD on this, and we still don't know how to do that. And but if you, so when you look at the genome, so this actually was work from Sylvie de Ken lab about 10 years, 12 years ago, 13 years ago now. They saw the first segment of about 65 kb, 20 genes coming from Torula spora, they name as region C. And basically, if you look at this segment of 65 kb, you see this segment into Torula spora genome. And it's only present in some of the Cerevisiae genomes, it's not present in Paradox, and so we know the direction is that one. But one thing, so we should be working quite a lot recently on this. So one thing that's emerged from the 1011 is when we sequence the 1011 strains, we did see other strains like this one, the palm wine, that actually has a longer segment. And actually there was a single strain from the Casberg collection where this actually we were able to extend to 165 kb, the original segment of 65 kb. Now again, if we take this 165, we can map to the same place in the Torula spora genome. So what happened is we think this one is what is the closest to the ancestral event. We don't know if there could be, it could have been even larger, but what has occurred is sort of reducing dimension, has been sort of deleted in the different strains. I think this might reflect the fact that it's costly and perhaps deleterious to have a segment of the genome that is highly diverse compared to the rest. And I think there are evidence where showing that what has remained in the wine genome, it might have been actually adaptive for fermentative conditions. So there are some nice data showing that this is the case for some amino acid import and during wine fermentation. One of the latest things we found, actually, we do see telomeric repeats at the end of this segment coming from the Torula spora genome. So I always get the question, why do we see this segment at telomeres? I think there might be more than one answer. So one is can be tolerated better by the host genome because it doesn't impair segregation, doesn't disrupt important material. And now we do see actually telomeric sequence from Torula spora showing that this might be needed to sort of stabilize this HGT at the beginning. This is the HGT. This instead is its introgressions. That's why I think these are two distinct mechanisms. So these are a group of strains characteristic of olive oil production, which is this one after centrifugation. So there is quite an amount of solid part, and it's rich in nutrients, both in metals and lots of other things like polyphenols. And so this group of strains is called alpecin, which is what this liquid is called in Spanish. And they come mainly from Spain, but now we have a large collection of this alpecin from also Italy, Slovenia, and other places. If you look at this genome, so this is the most introgress clades. So this is about 5–6% of their genomes have paradoxus ancestry. And these blocks that you see here, they essentially contain the counterpart of genes but from paradoxus. So one thing actually we did find when we're sequencing the genome, we did see actually something which we call the living ancestor. So this was an hybrid between Cerevisiae and paradoxus, coming from this olive oil wastewater. And what we did see this, so this is basically this hybrid has a complete genome from Cerevisiae and paradoxus, but also we do see these red blocks into the Cerevisiae genome. So these are called loss of heterozygosity, and these are again scattered throughout the genome. And Melania and other people in the lab have worked for many years on this. And one striking thing that you can easily see here is when we look at the position of these LOH blocks in the hybrid is largely overlap where we see introgression. So this has been sort of a question that we try to answer for many years, because when we have this hybrid, essentially it's sterile, so most of the gametes are dead, and it was sort of hard to understand this introgression or spread from these hybrids. So basically, we propose a new model where the hybridization. So the hybrid is perfectly fine. You can hybridize Cerevisian paradox, for example, in the lab. You can make new hybrids. This, if you propagate them methodically, they are perfectly fine. But the gametes are dead. But what we did also experimentally show is if you have this genome instability that creates this loss of heterozygosity, now these blocks of sequence identity can mediate recombination. And basically what we did show is we're able to rescue part of the gametes viability, and then we can explain why we do see this in the Alpecin lineage through the backcrossing and things. So this is where we place our hybrid that has essentially evolved in parallel to this to the lineage of the Alpecin that has backcrossed to the Cerevisiae, and sort of has revealed how this has occurred, or at least partially revealed.
So this was done in the lab? So
in the lab, essentially, we use this living ancestor, and experimentally, we have shown how we show experimentally the effect of these LOH blocks in recombination, both mitotic and meiotic. So basically, in this work, we combine both the population genomics of the Alpecin, reconstruct the living ancestor genome with experiments that were sort of initiated from this to try to understand how this has occurred.
So you didn't start from the hybrid itself?
So we've done many experiments with this. We don't have this. Essentially, this doesn't exist anymore. Mm. We essentially have compared experiments made year and year. So, for example, if we can generate gametes from this one, and then study the recombination and the effect of this loss of heterozygosity blocks.
And this genome instability, where do you think it comes from?
So we don't know. We're actually working on this right now. So what we know is if we take these and evolve mitotically, the hybrid is established. This is strongly inhibited. These LOH events, they are strongly depleted compared if you do this hybrid between two Cerevisiae strains, and this is due to mismatch repair and inhibition of homologous recombination. So we think this has been something transient, maybe driven by the environment or occurred during the early phase of hybridization. These are the two things we try to explore at the moment.
So for some reason, in some certain stress condition, this was actually selected for.
Yeah. So one things we tried is it's something that is called essentially. One things we've been trying to explore is something called aborted meiosis. So basically, we induce sporulation, but before commitment point, we go back to normal growth. But this has already sort of induced double-strand breaks in the genomes, and which need to be repaired. So this works very well for interspecies hybrids, but again, we do see strong depletion of LOH formation in the Cerevisiae paradoxal hybrids. So essentially, we don't know how this ever occurs. So one thing we explore is a different environment whether there could be also, I don't know, a genetic and environmental combination of things have promote such genomic instability. But we never seen anything similar to what we observe as occur here what we call the living ancestor. So in here we do find like 100 blocks of loss of heterozygosity. For example, when we do the retard to grow, we observe like 3-4 in the genome. It could be that, I don't know, there'd be multiple of these cycles, but yeah, we don't know. So just to wrap up this part, and maybe I will also conclude here. So I think our study has shown the sort of origin of the species in Southeast Asia, in several islands like Hainan and Taiwan, but also inland China. And this has been confirmed by groups of Feng Yanbai and Jason Tsai recently. We showed that there's been a single out of China, and then there's been sort of dispersal. This has sort of established a wide population of yeast in a number of environments, mostly are temperate forest, also sometimes is other type of forest. Although most of the Saccharomyces cerevisiae wide population that live in forest are found mostly in the North Hemisphere. So we hardly seen native wild population in the South Hemisphere, maybe except Malaysia and more in the sort of rainforest and in Ecuador. But I think this reflects probably the origin and the dispersal of the species. Then we do see multiple domestication events. This is probably has occurred multiple times. At least 2 independent domestication events are the wine type of domestication and the sake. They're very different ancestry, and this is occur independently. Also, there are a number of domestication events probably occur in China, reflected in sort of artisanal fermentation and baking process. This has also resulted in a further dispersal and redomestication. So basically, you do see a number of things like, for example, I don't know, we have wild isolates in Madagascar, but when we sequence them, basically we do see we can reconduct mostly to wild lineage that have been isolated, and these were sort of a mixed population. I think we did show that also there's been at least 2 independent hybridization and integration events, the one we did see in the Alpecin and these olive oil related strains. But also there is a number of American lineage of Cerevisiae that have integration from American Paradoxus. So we know that this at least has occurred 2 independent times. Many are characteristic of agave fermentation in Mexico or bioethanol production. And I think so basically population genomics has helped to illustrate many of the key events that have sort of defined the genome of the species. So we have an ongoing project with Jill Fisher and Joseph Schorscherer on 150 genomes. So we have probably the first panel of many genomes where all the chromosomes being assembled de novo from telomere to telomere. So we are building a new level, a new layer of genetic variation. There are new approaches to project genetic variation, for example, exploiting pangenome graph. This is work from Lorenzo Tattini and my team. Where basically try to reduce the fact that we need to project data into a reference genome, and instead you can compare genomes without the needs of a reference. And also we are currently isolating hundreds of strains from thousands of samples. So when you take wild samples, usually you have a success rate of isolating Saccharomyces between 10 and 20%. So these are a number of sites we have explored nearby us, but we are collecting samples from many different countries and continents. So I will skip, given the time, I will skip the second part, which it was to look at So essentially, we look at standing variation in this part of, referring to yeast. We also have approaches to look at de novo variation, which will mimic sort of the clonal evolution, and this can be done at very with high precision in yeast, because we have like yeast and bacteria, you can have access to sort of fossil record where you can sort of freeze your experimental evolution, go back to it. You can isolate individual clones and sequence them and phenotypes. So this has been highly powerful, but I think.
So Dan, if you want, I mean, there is no. It's really up to you. If you're tired, we can stop. But if there is more time, we can.
I think. Well, I can just quickly say what we find essentially without really going to the experimental details.
Yes.
So we essentially set up a system where we can compare the standing variation that has accumulated over a long time frame, like the one that we do see segregating in the 2011 yeast collection, and the novo variation where instead we do that accumulate during sort of laboratory experiments, timescales. So we use, as a system, we use resistance to a drug, in this case rapamycin, and we look at what it does contribute to resistance to rapamycin by mapping standing variants versus de novo variation. So first of all, we do see at the gene level, we do see the same target genes. So for example, we do see genes involved in the TOR pathway that get mutated during the experimental evolution. And also we do see natural variation in these genes that contribute to the same resistance.
Sorry, Jean, for standing variation, now you mean that you don't start from clones, but you have a...
Yeah, sorry. So standing variation is essentially the pre-existing variation in the population. So it's the one that originates during the evolutionary time of the species. So whereas the
mixed natural population.
Yes, exactly. Yeah. So in this case, if we take, I don't know, a Y strain and the Saki strain, so the difference between the two of them will be the standing variation, and then if use them through genetic crosses and so on and generate new variation, we'll call like de novo variation.
Mm-hmm.
So there is some overlap, but actually what we do see is that the mechanism of drug resistance was different. So in the de novo variation, they acquire few very strongly adaptive mutations that will essentially avoid the drug, in this case was rapamycin, to bind to the TOR pathway. Whereas the standing variation, it doesn't, we don't see the same mutations in the same part of the protein. Instead, we do see that change the sort of the activity of the TOR. So basically the variants that give more resistance, they give more TOR activity, and basically this was really the first system where we can compare the two types of mutations and basically as quantitative assess their adaptive effects, and also to ask whether there was a cost of having them or not. Yeah. So just to finish, to thank the team that has been working on various projects, and also we are a very collaborative research group, and over the years I collaborate with many researchers for a number of different questions.
Great, thanks a lot. Really, it was very, very nice. Are there questions from anybody? In the meanwhile, I ask, so the conclusion from the last point is that in laboratory evolution doesn't really mimic evolution.
So it's extremely hard to come up with a good experiment to sort of test, for example, even the fitness effect of something that we map in natural populations and to try to understand what it does. A lot of time we, I mean, not just us, but what we look at, for example, whether it does have an effect on growth rates under specific conditions. And instead, I think, so what we've been trying to do is it's also try to test these genetic variants across an ensemble of phenotypes, not necessarily how fast they grow. So we try to measure them through a number of phenotypes that could sort of illustrate and recapture the life cycle of the yeast. And for example, I don't know, also the ability to the reproductive capability of the cells, or for example, the fact that whether they can survive and so on. But it's always hard to set up good experiments to interrogate, let's say, the adaptive value of a variant.
And the ploidy of this yeast that changes, or the number of chromosomes is a constant?
Yeah. So in wild, we do see that most